It is closed.
Open. You are in the house live, sir.
Oh. Hi, good morning. Good morning once again to all of you. Now we are going to have a 4 days discussion on some topics in class 12th related to the subject biology. Right. And we will try to discuss some of the major things happening, major things in class 12th textbook only. And in between, we will discuss some questions also. If you have any doubts anywhere, please keep it in your mind and put it in the chat box so that we will able to discuss your doubts later in very detail. Now, we are going to discuss the first topic, that is what the first unit of class 12th, reproduction. All of you know that reproduction is one of the major event in living organisms. It is one of the most important characteristic feature of the living organism, and it is not possible to live or exist the life without reproduction. Every living organism go for reproduction. Whereas a lower form of individuals normally go for a reproduction asexually only, asexual reproduction. See, asexual reproduction is having lot of disadvantages. The main disadvantage of the asexual mode of reproduction is lack of variations in the coming generations. If no variation in the coming generation, it is not possible to exist with in that changing environment, because our environment or the physical features of the environment is keep on changing all the time. If you want to cope the body with the changing environment, we should make variations in new generation. That is why most of all advanced individuals are going for sexual mode of reproduction, including the plants and animals. You know, the higher level of plants like angiosperms, they are always going for the sexual mode of reproduction only, because they have a special organ designed for that, and we call it as what? The flowers. Flowers are called as what? The sex organ in case of plants. And higher level of animal also going for the sexual reproduction only, and that we will discuss after discussing in detail about the sexual reproduction in angiosperms or these flowering plants. So let us see the topic, sexual reproduction in flowering plants. As I told you, flowers are the real reproductive organ in a plant, particularly the angiosperms. And this flower is designed exclusively for the sexual reproduction. And most of the plants are producing flowers with two types of sex organs, both male sex organ and female sex organ. Just to go through the picture given here on the board, it is a fascinating organ and consists of mainly four whorls. And these are the four whorls of a flower. The outer whorl is called as what? The calyx. Outermost one, it is given in green in color. Then second one is called as what? The corolla. And third one is what? The male reproductive part called as androecium. And the innermost and the most important part is called as what? The gynoecium. That is what? The real female reproductive part. Right. So now we will see its arrangement in details in a flower. See, this is what? The outermost one, calyx, and it is made up of what? The units called as what? The sepal. And here you can see the sepals. There are five sepal here as a wall externally. That is what? The outermost wall, we call it as collectively the calyx. and there is a world inside, and most of the cases it is very colorful, and it is mainly to attract the pollinators to do the pollination only. And this second layer is called as what? The corolla. And this corolla is made up of petals. And there are 5 petals we can see here. It is Pendapetalous flower only. 5 petals are there. And inner to this petal, there is another word. And actually, this is what the male reproductive part, and it is made with a stamen. It is made with a stamen only. And this stamen consists of, there are 2 parts. One is anther. This is what the most important part, produce what the male reproductive gamete, and this is what the filament, a small elongated structure that only connect anther to the flowers, right? And these are the 4 important words. Every word is having its own significance. And the first 2 words, that is calyx and corolla, these are called as what? The accessory words only, because it is not directly involved in the sexual reproduction. They are not producing any gamete. And whereas the innermost words, like what you can say, andricium and gynoecium, and these 2 words are directly involved in the sexual reproduction by producing male reproductive gamete called as pollen grain, and female reproductive gamete called as what? The ovules. So we will see more detail about this different worlds of the flower. You get the picture here. You can see all the 4 worlds. The outermost whorl is made with the sepal, and it's called as what? The calyx. And the next whorl is made with the petal, and it is called as what? The corolla. And it's the most attracting part of a flower. And innermost whorls are reproductive whorls only. It includes 2 things. One is what? Andricium, the real male reproductive part, and the innermost one, that is called as what? The pistil. You can see here, pistil exactly at the center, and we call it as what? The gynoecium, the real female reproductive part. And this organ present in a plant, angiosperm, only for the reproduction, sexual reproduction. That is why we call these flowers as what? The reproductive organ of angiosperm. And you can see all these parts of a flower one by one, and these are all what? The outermost whorl, that is a calyx only. Here you can see the calyx, that green colored one. It is normally covering the entire flower at the time of bud, and later after blooming, it will be there at the bottom as what? The outermost whorl. And it is what? The corolla, most attractive part. As I told you, normally it is very colorful, particularly the day blooming flowers. It is exclusively to attract the insect like pollinators only for pollination. Whereas the night blooming flowers, normally it looks what? Very white in color, because it is what? The contrasting color of the dark. That is why it can attract what? The moth like nocturnal insects for pollination. Then this is what? The andricium, the real male reproductive part. And this andricium is what you can say, stamen only, and it is made up of 2 things. One is anther, and another one is a filament. And anther is the very important part in case of stamen, because anther only producing male reproductive gamete called as what? The pollen grains, right? Then it is the innermost part of the flower, and it is what? The female reproductive part called the gynoecium or pistil, and it includes mainly 3 parts. This is what? The stigma region, where these pollen grains fall, or pollination take place in the stigma region only. And this is what? The style. We will see the significance of what? The stigma and style later. And the most important part of this gynoecium is what? The ovary. And this ovary only produce the female gamete called as what? The ovules. Then let us see the pre-fertilization events. Pre-fertilization events means what? The events happening before fertilization, right? And it includes what? Many aspects. One is what? The gametogenesis. Gametogenesis. Gametogenesis means what? The process to produce gametes, male gamete and female gamete. The process to produce male gamete is called as what? The microsporogenesis. And megasporogenesis is what? The process through which plants are producing what? The megaspore, that is what? The female reproductive gamete. So this is what the first event come under the free fertilization events. One is what you can say gametogenesis, only the process of formation of gametes, male gamete and female gamete. Formation of male gamete is scientifically termed as microsporogenesis, and female reproductive gamete produced through this megasporogenesis only. And microsporogenesis in the male reproductive part of the flower, remember, and the megasporogenesis in the female reproductive part, particularly in the ovary only. Then second event, we will see later in detail. And second event of the pre-fertilization process is what? Gamete transfer, that we call it as what? The pollination. Pollination, transfer of pollen grain from the site of origin to the stigma region. And this process is called as what? The pollination. And this can be of different kinds. You have to specifically by heart the technical term for different kinds of pollination. Self-pollination, there are two type of self-pollination in commonly in plant. One is what? Autogamy. Autogamy means it is happening in the same flower. Pollen grain produced by a flower, and the pollen grain will stick on the stigma region of the same flower, because here pollen grain and ovum both are getting matured at the same time, so that it is possible to stick the pollen grains to the stigma region of the same flower. That only we call it as what? The autogamy. It is what? A self-pollination only, but it is happening in the same flower. So we call it as what? The autogamy. Whereas what? The different flower of the same plant, that is also possible. After producing the pollen grain by a flower, it can be taken by a pollinating agent to the stigma region of another flower of the same plant. And that is called as what? The geotogamy, right? These two technical terms you have to by heart, because normally, particularly in multiple choice, chances are there to ask such kind of questions, right? That autogamy and geotogamy, both are self-pollination only, but the autogamy happening in the same flower, whereas the geotogamy in the different flower of the same plant. Remember always. When come to the xenogamy, xenogamy is what? The cross-pollination only. Cross-pollination means what? The pollen grain of a flower stick to the stigma region of the flower produced by another plant of the same species, but another plant, right? And such a type of pollination is called as what? The xenogamy or cross-pollination. Here, plants are same species only. The plants are belongs to the same species only, but they are what you can say going for transferring the pollen grain from one flower to another, required some kinds of agent for pollination. Even for geotogamy, pollinating agents are required. Whereas in autogamy, sometimes it is happening without even the pollinating agents, right? Then let us see what the agents helping for pollination. Because one flower is producing pollen grain, another flower is producing what? The female reproductive gamete. So to make this female gamete fertilize, required what you can say some kind of agents to transfer it. And such a type of agent helping for transferring the pollen grain from one flower to another is called as what? The pollinating agents. And these agents can be biotic agent or it can be abiotic agent. Biotic agent means what? Living agents like butterflies or honeybees or moth or bat there are so many, even birds can also helping for this pollination, and such a type of agents, living agents for pollination is called as what? The biotic agents only. And abiotic agent means mostly wind, water. For example, paddy, it is transferring its pollen grain through the wind only. And aquatic plants, like Vallisneria, it is pollinating, it is getting pollinated through this water only. So these are abiotic, non-living agent helping for pollination. And some technical terms are also mentioned here. Note down the technical term, this anemophily. Anemophily means what? The agent is animal only. And hydrophily, here agent is abiotic agent, that is water only. And endomophily, here insect only acting as a pollinating agent, like a butterfly, honeybees, etc. Then ornithophily, birds, you may be heard about the hummingbirds, can do pollination, and such a type of pollination by using these birds are called as what? Ornithophily. And chiropterophily, that is what? The bat acting here as what? An agent, right? So anemophily, hydrophily, endomophily, ornithophily, and chiropterophily. And these are all what? The technical term related to the biotic agents only. And if agents are what you can say animal, we call it as what? Anemophilic. And hydrophilic flower means what? The flower using what? Water as a pollinating agent. And endomophilic flower means what? The flower using insect as their pollinating agent. And ornithophilic means they're hummingbirds, like birds only used for pollinating, transfer of pollen grain from the site of origin to the stigma region of some other flower belongs to the same species. And chiropterophily, and here bat is acting as what? The pollinating agent. Then let us see the detail of the microsporogenesis. What is microsporogenesis? I told you initially it is what? The process to produce male reproductive gamete. Male reproductive gamete produced in the male reproductive part of a flower, like anther. And in anther only these microspores are produced. Microspore means what? The pollen grain, the real male reproductive gamete. And this process to produce male reproductive gamete is called as what? The microsporogenesis. And it is produced from the sporogenous tissues. Sporogenous takes tissues, and it is present in the anther, the real male reproductive part of a flower. Right. There are many cells in anther other than sporogenous tissues for the nourishment of pollen grain and also for protection. So we will see the detailed structure of anther. Then we will find out what the sporogenous tissues. And the sporogenous tissues are the real reproductive tissues in anther to produce male reproductive gamete, called as what? The microspore or these pollen grains. And see the structure of an anther. This is what anther. I told you collectively we call it as what? The stamen only, the male reproductive part consists of anther and filament. And this is what? The cross section of the anther. And these pollen grains or these spores are produced inside this anther only. And after the production and maturation, it will go for what you can say breaking longitudinally, and entire pollen grain present inside this anther will come out and stick on the surface. Whenever this insect like a pollinating agent sit on a flower for getting its sweet liquid, we call it as what? The nectar or honey. And this pollen grains, its structure is different, having so many spiny things on its surface. And the body of insect also designed for this pollination only. And this will stick on its body and taken to some other flower. And it is happening accidentally. It is not a purposeful action by the insects. And this is what the microsporangia, and this only developed to become the pollen sacs. It is what the transverse section of an anther. Transverse section of anther, you can see that the bilobed anther only, and this is what the transverse section of the anther. And this is what the microsporangia, structure of the microsporangia. As I told you, there are so many tissues present in the microsporangium, and outer layer, it is made up of what? Epidermis, endothelium, and this middle layer. There are three layers present in the outermost layer of this microsporangium, right? Microsporangium. This is what the microsporangium in which that pollen grains are getting developed, right? And these outer layers are epidermis, endothelium, and this middle layer. And the core layer is what? The microspore mother cell. I told you about the microspore mother cell, and this microspore mother cell only responsible for producing microspores. And the different kinds of tissues present in the microsporangium are for gamete production or microspore production. Then some are for nourishment and some are for protection, right? And this outer layer is exclusively for protection only, like epidermis, endothelium, and middle layer of this microsporangia is for its protection, whereas this core area we can find the microspore mother cell, and this microspore mother cell only responsible for producing the microspore or male reproductive gametes, right? Then here we can see the inner layer, that is called as what? The tapetum. Tapetum is the innermost layer, and it is exclusively for nourishing the cell, particularly for the development of the pollen grain from the microspore mother cell only. It is for providing the nutrient. It is for the nourishment. Same question asked in the pre-board examination. Also in 1 mark question, it is asked, right? Which cell present in the microsporangium nourishes the microspore during its development? That is what? The tapetum only, right? And by using tapetum, this get matured, and after the maturation of the pollen grain produced from the microspore mother cell, it break its wall, that middle layer, endothelium, and epidermis, that go for breaking, and everything will come outside and attach to the surface of the anther region, right? That only taken by the insects. As I told you here, these three areas. Cell present in a microsporangium. There are 5 layers present in the microsporangium, and it is in 3 sections only: outer 3 layer, inner layer, and center of the microsporangium, right? The outer layer, it is exclusively for protection and dehiscence, and it includes 3 type of cell. Epidermis is there, endothelium is there, and middle layer is there, right? See, this is what? The epidermisal, endothelium, and middle layer. These 3 layers are called as what? The outer layer of what? The microsporangium. Then inner layer, it to nourish the developing pollen grains only, and it includes what? The tapetum. And it is multinucleated cells only. This tapetum is multinucleated cell only, and the nucleus is also very large in size, and it is exclusively for nourishing, or it is exclusively for providing nutrient to what you can say the microsporangium mother cell to produce what? Or to provide the sporogenous cells to produce the microspores. Right, that is what the inner layer to nourish the developing pollen grains. Here you can see what the inner layer, that is what? The tapetum only, and it is multinucleated, and normally nucleus is very large and spherical in shape, and exclusively for nourishing these male reproductive gametes only during its formation. Then come to the next what you can say center of microsporangium, and that only helping for the development of pollen grain. Spermatogenesis, I mean this microsporogenesis. That microsporogenesis is happening from this, what you can say, this sporogenous mother cell only. And this sporogenous mother cell, you can find it here. This is what? The core microspore mother cell, and this microspore mother cell only developed to become the real male reproductive gamete called as what the pollen grains. Right. These are the cell present in the microsporangium. There are 5 layers, and outer layer is made up of 3 kinds of cells in 3 layers only. Epidermis, the outermost one, next endothecium, and next one is what the middle layer, and the inner layer is tapetum for the nourishment of the pollen grains only during its development as well as the maturation, and that center of microsporangium, and it is made up of microspore mother cell, and this cell is only responsible for producing microspore or male reproductive gametes. Then microsporogenesis. Let us see the process of microsporogenesis. Microsporogenesis means what? The process to produce male reproductive gametes. So let us see, this topic is also very important as far as your examination is concerned. Not only the microsporogenesis, megasporogenesis is also equally important, right? So you have to explain the process of microsporogenesis with a schematic diagram. So easy to understand, easy to by heart, and easy to explain the process of formation of microspore from microspore mother cell, and this process only we call it as what? The microsporogenesis. And it is, as I told you, microspores are always produced from the sporogenous tissues only. Right. And this microspore, sporogenous tissues are called as what? The microsporocytes. Microsporocyte. And these microsporocytes are capable to produce microspore mother cell, MMC, through the process of mitosis. You remember, these cells are diploid in nature, but microspores are haploid. So the process of production of haploid cell from the diploid cell only we are going to discuss here in case of what? The microsporogenesis. This is what the real process to produce a haploid cell from a diploid cells only. And actually, the sporogenous tissues present in the anther only produce what? The microspore mother cell simply through the mitosis. Mitosis means what? The equational division only. So naturally, the microspore mother cell also will be a diploid cell only. And microspore mother cell produce microspore tetrad through another type of division. We call it as what? The meiosis. So there are 2 types of cell division involved in the process of what? The microsporogenesis. One is what? The simple mitosis or equational division, and another one is what? The reduction division, or we call it as what? The meiosis. And mitosis take place in what you can say microsporocyte or the sporogenous tissues only, and that only produce what? The microspore mother cell. Naturally, microspore mother cell also diploid in nature, and this diploid microspore mother cell undergo second type of division. We call it as what? The meiosis or reduction division, and that only produce what? The microspore tetrad. You may be remembering the process of meiosis. Whenever a diploid cell undergo the process of meiosis, in meiosis there are 2 division. Division 1 is what? The reduction division, and second one is what? The what you can say equational division only. So there are 4 cell produced due to this meiosis. That is why here microspore tetrad is produced from the microspore mother cell. Every microspore mother cell can produce 4 cell, and all these 4 cells are haploid because here division is what? A simple meiosis, or we call it as what? The reduction division. Why it is called as reduction division? Because during this division, what happened? The chromosome number reduced to half. Right. That is what? The haploid cell only will be produced at the end. Right. Then microtetrad dehydrate during maturation process and dissociate from the pollen grains. So this is what the what you can say overall things related to the sporogenesis. And the sporogenesis is what the process of producing the pollen grains, and these pollen grains are produced from the sporogenous tissues only, basically present in the anther. And these sporogenous tissues first undergo the process of mitosis and produce what the microspore mother cell. And this microspore mother cell again go for meiotic division and produce what a haploid tetrad, and this haploid tetrad will be separated, and that only develop to become what the real male reproductive gamete called as what the pollen grain or the microspore, right? And this is what the thing we discussed earlier, right? This is what the microspore mother cell. This is what the microspore mother cell for the development of the pollen grain, and the tapetum for the nourishment for to this what the microspore, and these are all for the protection only, right? And this microspore mother cell first undergo a mitosis and produce what the microspore mother cell undergo the process of meiosis because it is diploid and produce what tetrad. Tetrad means four cell, and all these four cells are haploid, and that only later develop to become what these pollen grains, right? Then this is what the schematic diagram showing the spermatogenesis. You by heart as it is, so that it is very easy to explain. Right, this schematic diagram or spermatogenesis. Whenever such question asked in the examination, you have to explain with the schematic diagram only. So it is very easy to explain, very easy to understand, and very easy to what you can say conclude the process, because the evaluators after seeing the schematic diagram will understand everything, right? So whenever such a question asked for your examination, definitely you have to draw the schematic diagram showing the spermatogenesis and explain its process, right? And I told you the spermatogenous cell, or we call it as what the microsporocyte. Basically, it is diploid cells only. These are all diploid cells only. Sporogenous cells are diploid cells only, and this will go for mitosis repeatedly. Mitosis means what? Equational division. So naturally it produces what? Many cell, all cells are diploid because mother cell is what? Diploid. Here mother cell is what? The sporogenous cells only. It is already diploid. So naturally that microspore mother cell produced through the mitosis also will get what? The same number of chromosomes. They are also what? Diploid in nature. Right. And this diploid microspore mother cell, again, it go for the next division that is called as what? Meiosis. In meiosis, there are two divisions. As I told you, the first division is a reduction division, reduce the chromosome number to half. And the first meiotic division, it produces two cells, and these two cells are haploid only. And whenever these two haploid cells undergo the next division, and next division is what? Equation division only. There is no further reduction in the number of chromosome in the section division, and it is what? Equational division only, simple mitosis only. So naturally, at the end, there are four cells produced here. And it is called as what? The microspore tetrad. Every cell is having only n number of chromosome, or what you can say haploid. They are all haploid in nature. Then later it disintegrated and separated, and this will be converted into microspore. That is the real male reproductive gamete, a functional reproductive gamete, and we call it as what? The pollen grains. So this is what the schematic diagram. You go through it. Sporogenous cells are the microsporocytes, and that only go for mitosis or equational division and produce microspore mother cell, MMC. And this microspore mother cell undergo the process of meiosis, and it is what? The reduction division only, and produce what? The four cell, we call it as what? The microspore tetrad. And microspore tetrad are all haploid, having only n number of chromosomes, and that will be separated and function as what? The male reproductive gamete. We call it as what? The microspore, or we call it as what? The pollen grains. And this is what? The functional pollen grain. microspore cells, right? It go for mitosis and it produce what the pollen grain. And this pollen grain consists of two cells inside. One is vegetative cell having n number of chromosome and also having what a generative cell and also having n number of chromosome. As I told you, the tetrad is what the haploid only, and whenever this the nucleus of the tetrad undergo the process of mitosis, it produces two haploid cell inside. Only the nucleus will divide. Only the nucleus will divide. So naturally, what happened? They produce two type of nucleus inside the same cell, and we call it as what the vegetative cell, and another one is what the generative cells. Right? That the tetrad is what you can say, four cells will be there, all four cells are haploid only, and every cell from the tetrad, only the nucleus will go for division further, that is mitotic or equational division only. And whenever this nucleus divide, it produce two nucleus. One is larger and we call it as what the vegetative cell, and another one is small and we call it as what the generative cell. So there will be two nucleus or two cell in a pollen grain. And this is what the detailed structure of the pollen grains. This pollen grain required protection because once it is exposed, the climatic condition is maybe very adverse. So it required protection. That is why it is protected with a type of structure here, and we call it as what the exine. And this exine is made of what the hardest non-plant material. We call it as what the sporopollenin. Sporopollenin only present in this what the exine, and it is exclusively for the protection only. And there is a structure inner to that that we call it as what the intine. And here you can see a place where there is no exine cover. And this structure only, this part of this pollen grain only, we call it as what the germ pore. Germ pore is what the area where there is no exine cover. Only one or two such germ pore present in a pollen grain, whereas all other areas of this pollen grain is protected with what you can say the hardest non-plant material, and its name is what the exine. The name of the material present in the exine is what the sporopollenin. One this question also asked in the board examination. And sporopollenin, it is the hardest non-plant material. With this material only this exine is made, right? So there is a structure inside that is called as what intine, and in germ pore area that intine is directly exposed to outside, and entire material present in the pollen grain is taken outside through what we can say that germ pore only, because all other areas are very hard and very impermeable and cannot take anything outside, and it is exclusively for protection. Once this pollen grain is released, and it should be what you can say stable in the awkward climatic condition. That is why it is covered with such type of hardest material, that we call it as what the exine cover only, right? Excuse me 1 minute. Excuse me 1 minute.
Click on the button. I think the you zoomed in. Click on that, your end size slide shows.
Excuse me for a minute. Technical error. Correct it now. Yeah.
How can you check? Yeah.
What happened?
Which slide was it?
The microsporogenesis after this one.
Just from with one finger. was it?
But it is.
Two fingers.
Okay.
Wait. Is it fine if you click from here?
From here, huh?
This button.
This will.
Button next. This is the previous.
Yeah, yeah.
Okay.
Yeah.
I see after the
Okay,
lecture.
okay. Okay, thank you. Yeah. See, this is what the things we discussed now. The detailed structure of functional pollen grains. After producing this microspore cell, and it is a haploid only having a single nucleus, and this is what haploid cells only. And this haploid cell undergo, not cell, the nucleus undergo the mitotic division and produce two nucleus. Only the nucleus will go for the division, and division is mitosis. Already it is haploid, so whenever it undergo for the equational division, it produce two haploid nucleus inside the same cell. And one nucleus is larger, we call it as what, the vegetative cell, and another one is smaller, we call it as what, the generative cell. And later stage, it undergo the process of some kinds of development by producing what, an external cover for protection, we call it as what, the exine. And this structure only acting as what, the intine. Then exine is covering almost all the area except one or two small area, and this area is directly exposed through what you can say intine, and we call it as what, the germ pore. But at the time of what, it's further even the materials are taken through that germ pore only. And come to what, the general characteristic features of what, these pollen grains. It is normally spherical in shape, very small and microscopic, and its size is what, ranging from 25 to 30 micrometers only. And there is a very hard outer layer, as I told you, it is called as what, the exine, made up of what, sporopollen, and it is the hardest non-organic matter or plant material. And exine has a prominent aperture, that is called as what, the germ pore. In germ pore, there is no exine cover. It is what, a small aperture where exine is not there. And inner wall is called as what, the intine, and it consists of two cell inside. One is generative cell and another one is vegetative cell. Generative cell and vegetative cell. Generative cell is larger, as I told you, and then what you can say, another cell present in the pollen grain called as what, the generative cells. Once again, you see the diagram of this pollen grain having two nuclei inside, vegetative nuclei and generative nuclei. Vegetative nuclei is larger in size than what you can say, the generative cell, right? Then come to these pollen grains. See, pollen grain can cause allergies and respiratory afflictions that we call generally as the pollen allergy. You maybe notice many people, particularly pollen grains, acacia produce pollen grains together and it will spread in the atmospheric air. And during this period, some people are getting some kinds of what the allergic response, like a difficulty in breathing or itching in their body, etc. And pollen grains are rich in nutrients. Pollen grains are rich in nutrient, and maybe heard about the pollen tablets and pollen syrups, and it's good for what you can say, very good food supplements only. It's full of nutrients, and viability of pollen grains in rice and wheat is less than 30 minutes. Viability means what the period in which it live, that is viability. And rice and wheat, it is less than 30 minutes, whereas in cases of leguminaceae, and its viability is more than 1 month, because it is having an outermost exine cover. It is able to live for 30 minutes, 30 days or 1 month. Then pollen can be stored in liquid nitrogen, that is what 196 degrees Celsius for many years. So what you can say, agriculture research fellows are storing this pollen grain for producing new varieties of plant by keeping it in liquid nitrogen for many years together. And pollen banks play important role in breeding program. As I told you, this pollen grain that is stored in the liquid nitrogen can be used in future to generate these varieties, different varieties of plants. And this is called as what the pollen bag, where we used to keep lot of pollen grains in a liquid nitrogen. Right, and this is what the structure of pollen grains. We already discussed in detail about the structure. This is what the germ pore, where there is no exine cover. And this is what the outermost cover of the pollen grain, we call it as what the exine. And this is what the layer called as what intine. And there are two cells present inside. One is larger and another one is smaller. The larger one is vegetative cell and smaller one is called as what the generative cell. This is what the part of the pollen grains, the internal arrangement of or internal things present in the pollen grains only. And this is what the outermost, what you can say, it is a look of what pollen grains outside. I told you many spiny things are there all over this pollen grain, that really helping the pollen grain to stick on the body of the pollinating agents like insects, right? Then see, insects, they are sitting on a flower for getting their nectar. Whenever they sit on the nectar, these are all yellow colored one is pollen grains only, matured pollen grains on the surface of this flower, male reproductive part, and that will stick on their body. And their body is also designed in such a way, having so many spiny things all over their body. Whenever it sit on a flower, naturally the pollen grain will stick on their body. And whenever this insect visit another flower for the same purpose, chances are there for this pollination, right? Then come to the next one, that is what the megasporogenesis. What is megasporogenesis? It is the process to produce female reproductive gamete. Process to produce female reproductive gamete. That is megasporogenesis. And megaspore is female gametes only, or we call it as what the ovules, right? So it is a process of formation of female gamete called the megaspore or ovule. And this question also asked in the pre-board examination for 5 marks, right? This megasporogenesis, and it is equally important because it is frequently asking in that board examination, that megasporogenesis, it is a process of formation of female gamete called as what the megaspore or ovule. A megaspore produced in female reproductive part of a flower called as what the pistil. Remembering the structure of the pistil, that is what the gynoecium, innermost part of the flower, that is what the female reproductive part called as what gynoecium or pistil. In pistil is made up of 3 areas, 3 parts, maybe remembering that the top free end is called as what the stigma region, then style region, and bottom round area is called as what? The ovary region. In that ovary only, the real process of what this megasporogenesis is happening. Right. And see the structure of the pistil. Pistil means what? The gynoecium only, female reproductive part of flower. And the same flower consists of a single pistil called as what? The monocarpellary, or we call it as what? The carpel. Is the pistil is otherwise called as what? The carpel. If only one pistil in a flower is called as what? Monocarpellary. And some flowers are having what? Many number of carpel in a flower. We call it as what? The multicarpellary. And sometimes pistil may be in fused condition, and such a fused many number of what carpel is called as what? The syncarpus. And sometimes all these multicarpus or number of carpel will be there in a flower. These are all not fused, these are all free, and we call it as what? The apocarpus type of flowers. Right. And this is what the structure of the pistil, female reproductive part of a flower. This is located exactly at the center. This is the part of the flower required a maximum protection, because this part only developed to become seed and other parts later after fertilization. We will come to know that. Then this is what the stigma region, that free end where the pollen grains come and stick. And this is what the style region, and through the style only this male reproductive content is reaching to the ovary. Ovary is located at the lowermost part. This is what? The ovary. In this ovary, this is what? Some flowers are like this only, stigma, style, and ovary. Right. And inside this ovary only, the real female reproductive gamete, we call it as what? Ovule, or the megaspore is getting developed. And this is what the detailed structure of megaspore. Now we are going to discuss in detail about the process through which megaspore is produced in the ovary by a flowering plant. That is called as what? The megasporogenesis. And these are the three parts of a pistil. This part is called as what? The stigma. Already we discussed about it. And this is what? The style. And the bottom portion is called as what? The ovary. And stigma only for pollination, and through style only it is passing, and finally it reaching to the ovary, where it meet what? This female reproductive gamete called as what? The ovules. Then come to the pistil, stigma, style, ovary. See, stigma is acting as what? The landing platform for the pollen grains. I told you that pollen grains are transferred by some agents like insect, and it is taken to what? The stigma region only. It is what? The landing platform for the pollen grains. Then style, it is elongated slender part beneath the stigma. Beneath the stigma, there is a slender elongated portion that is called as what? The style. And ovary is the basal bulged part of the pistil. The basal part is somewhat bulged, and that bulged part of what? The pistil present at the basal region is called as what? The pistil only. And ovary is the most important part. Among these three, ovary is the most important part of the pistil, in which ovules are developed. You know, ovule is the real female reproductive gamete, megaspore, that is getting developed in the ovary region only. Right. This is what the structure of this ovary. It is stigma, it is style, and this is what? The ovary. In this ovary, these ovules are located. See, ovule, it is what? The real female reproductive gamete, and it is located exactly in the ovule. Number of ovule varies according to the species, and some species like a mango having only one ovule in the ovary. Right. And it is ovule, female reproductive gamete, called as what? The megaspore. It is located exactly inside the ovary, and it is attached to the ovary with a structure here that is called as what? the funicule, and there is a cavity in which that ovule is located in the ovary, that is called as what? The ovarian cavity or locules. In the locules only it is getting developed and matured also in the locule present in the ovary only. Number of locules depend on what the species, some species of plants are having many number of ovule in the ovary. Every ovule is getting matured in different locules only. In case of mango, only one locule. In that locule only that megaspore or ovule is getting developed. Right. And this is what the megasporangium ovule, a megasporangium. So, as I told you, number of ovule present in the ovary varies from a single to many. In case of mango, only one. In case of hibiscus, we can find many megaspore or ovule in a single ovary. Single ovule, paddy, then wheat, mango, these are all having only a single ovule in an ovary. Whereas in case of papaya, watermelon, orchids, we can find many ovules. See, in case of mango, you can find only one seed in a fruit. After fertilization, that ovary only develop to become the fruits and the ovule develop to become its seeds. See, in case of mango, wheat, or paddy, only one seed in one fruit. That means you're having only a single ovule in it. And in case of papaya, you may be noticed, or in case of watermelon, you may be noticed many number of the seeds inside the fruit, because these many ovules are there in a single ovary, and every ovule develop to become what? Seed in the later stage. And this is what the detailed structure of the ovule, and this should be by hearted so that you can easily understand the process of what? The double fertilization. Always you have to explain the process of fertilization in the angiosperm by using the diagrams of these two gamete, male gamete, microspore, that is male gamete, and megaspore, that is a female gamete. We call it as what? The ovules. And this is what the structure of the megasporangium or the female reproductive gamete called as what? Ovules. Right. And here we will see detail also later, having two protective structure externally. These are the two protective structure, and we call it as what? The integument. And inside many cells are there, and these are called as what? The nucellus only. And nucellus are all diploid cells only. Integument is also for protection. And inside the integument, there is a structure that is called as what? The embryo sac. And inside this embryo sac, we can find there are 8 cells. Here, 3 are in this end. This end of what? The megasporangium is called as what? The chalacellan. And this end is called as what? The micropyle. And there is an opening here that is called as what? The micropyle. And this end is called as what? The micropyle. And this opposite end is called as what? The chalacellan. In an embryo sac, this is what? The embryo sac located exactly at the center. And in embryo sac, there are 3 cells in the chalacellan, that is called as what? The antipodal cell. And all these 3 antipodal cells are haploid cells only. And there are 2 nuclei you can find exactly at the center nucleus. And we call it as what? The polar nuclei. It is located exactly at the center. And some cases, these polar nuclei go for fusion and appears at the center as the center nuclei having diploid number of chromosomes. Otherwise, it is what? The haploid cells only. Polar nuclei also a haploid cells only. And there are 3 cells you can find in the micropyle end of this embryo sac. And there is a cell located exactly at the center, and it is called as what? The egg cell. And there are 2 cells on either side, and we call it as what? The synergids only. And both the synergids are haploid, and the centrally located egg cell is also haploid in nature. And this is what? The structure connected to the placenta of a flower. And from that placenta only is getting all the nutrient for its maturation and development. and it is called as what? The funiculus, right? We will see detailed structure later. That is what the funiculus, and this is what the ovule connected to this placenta of ovary. And there are two protective structure for this megasporangium or ovule. We call it as what? The integument only. And here there is no integument, and this small aperture where there is no integument cover is called as what? The micropyle. And we will see the significance of micropyle later. And this end is called as what? The micropyle, and opposite end is called as what? The chalasalan. And the most important part of the ovule is exactly located inside, and it is called as what? The embryo sac only. In this embryo sac we can find there are 8 haploid cells, and 3 are there in the chalasalan, called as what? The antipodal cell, and 3 are in micropyle, and centrally located cell in the micropyle is called as what? The egg cell, and on either side we can find the synergist. And there are two nuclei exactly at the center. Sometime it will be in the fused form only. If it is fused, it is diploid only. If it is not fused, this will be haploid only. And we call it as what? The central cell, or this polar nuclei, right? And this is what? The detailed structure of this megaspore, or female reproductive gamete. And we will see the details of the embryo sac. Embryo sac. This is embryo sac. Here you can find the embryo sac. This is what the detailed structure. It is given in blue color at the center of this ovule, and it is called as what? The embryo sac. There are 3 cells here. Here you can see there are 3 cells here. 3 cells are located in the chalasalan. 3 cells are located in the chalasalan here, right? Okay, here you can find 3 cells, and it is called as what? The antipodal cells only. And there are 2 nuclei you can find exactly at the center, that is called as what? The polar nuclei. And there are 3 cells in the micropyle. One is what you can say synergists on either side, and large cell at the center, we call it as what? The egg cell only, right? And this detail only mentioned here. One minute, excuse me, some technical issues. Sir, 1 minute, sir. 1 minute, same problem.
What's happening?
Oh, no, it is not moving actually.
Now can you try?
Yeah, now it's thank you. Yeah, come to the detailed structure of this embryo sac. It is located exactly at the center of an ovule. Here, inside the integument mass, I told you here the structure, this structure of the embryo sac only we are going to discuss here. This is what the structure of the embryo sac, exactly located at the center of an ovule. In embryo sac, we can find there are three cells in the chalasalent. Right, same thing. This is what the chalasalent. This structure only we are going to see in detail here. This is what the chalasalent. Three cells are there. We call it as what the antipodal cells. And there are three cells here in the micropyle end, and this is what the micropyle end of this embryo sac. There we can find three cells, and it is called as what the egg cell. Exactly at the center on either side we can find the synergids. These are all haploid cells only, and there is a nuclei exactly at the center, and it is called as what the polar nuclei. There are two polar nuclei, and these two polar nuclei go for fusion to form the central cell, and central cell is diploid because it is formed due to the fusion of two haploid polar nuclei only, right? And this only we are discussing here. See, inside the integument mass of cell called a nucellus, and this is what the nucellus, this is what the mass of cell present in the ovule, and these are all diploid cells only, and it is called as what the nucellus only. And the nucellus consists of rich amount of reserved nutrients. It is storing lot of food material. In the mass of nucellus, there is a large embryo sac developed from one nucellus cells only. So actually, at the time of what this formation, there will be no embryo sac inside, and this embryo sac is developed from any one of the nucellus. You know, nucellus cell is diploid cell only, and from this diploid cells only this nucellus, the embryo sac is getting developed. And the embryo sac is otherwise called as what the female gametophyte, and it is normally one in one ovule. Normally only one embryo sac present in one ovule, right? And this embryo sac is what the most important thing, and it is developed from any one of what the nucellus cells only, right? And this is what the embryo sac. You can find there are three cells in the chalasalent, that is antipodal cell. There are three cells in the micropyle end, two synergids and one egg cell, and there are two nuclei at the center called as what the polar nuclei. And last year's pre-board examination, this question asked the diagram of embryo sac. Whenever you are asked to draw the diagram of embryo sac, should not draw the diagram of ovule, you have to draw the diagram of this embryo sac only, showing what three cells in the chalasalent called as what the antipodal cells, and two are in a center, we call it as what the polar nuclei, and three are there in the micropyle end. These two on either side is called as what the synergids, and central located one is called as what the egg cells only, right? Then come to the process of formation of ovule, or that we call it as what megasporogenesis. Formation of megaspore, that is ovule, is called as what megasporogenesis, and it is produced from megaspore mother cell. You may be heard about microspore mother cell. From that microspore mother cells only tetrads are being developed, and similarly that ovules are developed from the megaspore mother cell, and this process is scientifically termed as what the megasporogenesis, formation of megaspore from megaspore mother cell. And development of embryo sac from a single megaspore is termed as what the monosporic development. This also asked once in the word examination, what do you mean by the term monosporic development? I told you about the what you can say development of the embryo sac. Embryo sac. Embryo
See,
sac normally
there is
only
concept of
contains
loss
one
of
ovule, and this embryo sac
or
is produced
more.
from or developed from a single
Maybe one or two.
what you can development of embryo sac
Loss
from
of
a single megaspore only, and it is termed as what the
gain of
megasporic
gain.
development.
Loss or
Then
gain of
see,
one
this
or
is our
more.
ovules are formed in the ovary
Loss
wall
of
as a small. Let us see what are
gain
the
of
events happening during the process
one or
of
more.
the megasporogenesis. What are the events happening during the
Right
process of what?
results. Results.
Megasporogenesis. Right. And ovules,
Failure
so
of segregation
the
of
ovary
homologous pair
wall
of chromosome during the
are a
meiosis.
small outgrowth from the placenta. It is formed. This is what the placenta, and this is called as what the locule in which what the ovule is getting developed. There is a space in the ovary, we call it as what the locule only. And first of all, these ovules are getting
Is
developed
given the
from
loss.
the
Okay,
placenta.
why there is a
A
loss?
small
We'll see later.
outgrowth.
Failure of segregation. Okay,
There is
failure
a
of
small
segregation
outgrowth
of homologous.
produced from what you
Pair
can say
of
placenta.
homologous
Placenta
pair
means
of
what? The
chromosomes.
structure present in the ovary which provide the nutrient for the development of what this ovule inside only, right? And inside the locule
Pair
only,
of
it is
these.
developed
Okay.
as a small outgrowth.
What is the reason for unemployment?
Let us see
Okay,
what happens.
that means usually you are deployed, right? There is a loss or gain.
Yeah. Then, actually, after the formation of small buds from this placenta,
There is a
the
loss
buds
of one
are
chromosome
and produce
from
what?
your
A
deployed.
multicellular
Suppose instead of
structure like
40,
this, and this
6,
cells
there is
only
a
we call it
45.
as what? The nucellus.
Monosomy.
And nucellus is actually diploid cells only, and this diploid cell, from this one of the nucellus cells only, the real embryo sac is getting developed, right? That only we are going to discuss here in detail. First of all, these ovules are formed from the placenta of an ovary as a small buds, and the cell present in the buds undergo the repeated division and produce what? A multicellular structure, and all the cell present in the buds are diploid cells only, and we call it as only. And the nucellus cell in later stage, it's rise up from the placenta and produce what? A structure called as what? The funicule only.
Only this much.
Right.
Many are
This
there.
bud slowly rise up from the
If it is
placental
the same
wall
type or a
with the
different
help
type,
of a
both are
stalk-like
achieved.
structure, and it is called as what? The funicule, and also increase its size by absorbing the nutrient from the placenta. From the placenta, this buds continuously absorb the nutrient and it's go for further
So
division and
aneuploidy is the
also
result of
for the
what?
rise
There is
up
a
from
loss
the
or gain of.
placenta only.
Okay.
Right. And
there is a loss or gain of one or more chromosome. That is the result due to the failure of segregation of homologous pair of chromosomes. Okay. It occurs when? Usually the occurs in the meiosis. Right?
1 minute.
Now, which stage in the meiosis this occurs? Huh? You एक मार के मेरे आंसर लास्ट में बोल.
Not this one, sir. That plant, no, this is some other topic. Flowers, reproduction in flowering plants, right? That will be. Here. Yeah. See, once again, let us see, there is a small break. So to get the continuity, let us move once again from the previous slides. Right. So here, now we are discussing about the formation of what? The ovule in the ovary, that is called as what? The megasporogenesis. Actually, that ovules are formed from the placenta of an ovary as a small buds only or as a small outgrowth only. And later, what happened that cells undergo the process of mitotic division and produce a multicellular structure. And this cell present in this that outgrowth formed from the placenta is called as what the nucellus. And here all the nucellus cells are diploid in nature because they are all produced through what the mitotic division only. And such a type of what the nucellus cells only later what you can say develop to become what the ovule. And after a period of cell division through mitosis, what happens slowly is rise up from the ovary and get a stalk-like structure that only we call it as what the funicule. We will see the role of the funicule later. And here this funicule is nothing but a stalk which connect this what the buds to the placenta of the ovary only. And later, what happened? This outgrowth is covered with one or two layers of structure. Here we can see the two-layered protective structure that is called as what the integument by leaving a small gap here that is called as what the micropyle. Micropyle is a small gap where there is no integument cover. We can find such only one area in an ovule where there is no integument cover, and this small pore aperture is called as what the micropyle only, right? And it is what the multicellular structure, and you can find so many nucellus cells inside, and it is covered with two layers of protective structure by leaving a small aperture here that is called as what the micropyle. And here one nucellus cell, it is what a multicellular structure, all the nucellus cells are diploid in nature, and one of the nucellus cell present in this structure what you can say converted into megaspore mother cell. Many are there, many nucellus cells are there, and one of the nucellus cell will be converted into megaspore mother cells. Now we are discussing in detail about the formation of the ovule in the ovary, that is called as what the megasporogenesis only, right? And here what happened, that cell is having a nucleus, megaspore mother cell is having a diploid nucleus, and this diploid nucleus undergo meiotic division, and it produce what the haploid cells. Haploid cell. This is what the MMC or megaspore mother cell, and it is diploid in nature. And such a diploid cell only undergo the division, meiosis 1, produce two mother cell dyad, and it is called as what the mother cell dyad. Two cell produce from a megaspore mother cell. Megaspore mother cell is nothing but a modified nucellus cell only, and this megaspore mother cell go for meiosis. I told you earlier, meiosis is having two division. One is what the reduction division, and the second one is what the normal equational division only. And first division is called as what the meiotic division number 1, and second one is called as what the meiotic division number 2. In meiotic division number 1, it reduce the chromosome number to half because it is reduction division only. So what happened, this diploid cell, diploid nuclei present in the megaspore cell will go for reduction division and produce two haploid cell, and we call it as what the dyad only, mother cell dyad. And this mother cell dyad again go for the next division, that is meiotic division number 2, that is what equational division only, and produce what these four mother cell tetrad. We call it as what the mother cell tetrad. Same thing we have seen in case of what microsporogenesis also, that tetrad. There are four cells, and all these four cells are haploid in nature because it is produced through the meiotic division only by the megaspore mother cell. Megaspore mother cell, and it produce what the mother cell dyad here, and this megaspore mother cell dyad again go for the second division, produce what the mother cell tetrad. The same thing, megaspore mother cell undergoes a meiotic division, that is a reduction division only, the first one, and produce for haploid cell and called mother spore tetrad, mother cell dyad, then mother cell tetrad. Finally, four cells will be produced, right? Then This is what the process of what this division here. First, this megaspore mother cell undergo the first division here. You can find many nuclear cells around it. These are all nuclear cell. One of the nuclear cell only converted into megaspore mother cell during the formation of the microspore megasporogenesis, and this megaspore mother cell is having a diploid nuclei here in the nucleus, and this nucleus only, all this megaspore mother cell only divide. The first division is what the reduction division and produce two cell megaspore dyad, and these two cell megaspore dyads are haploid cell, and this megaspore dyad again go for the next division, that is simple equational division only, and naturally it produce what the four cell here, and we call it as what the megaspore tetrad. So total four cell produced from the megaspore mother cell. Remember, megaspore mother cell is nothing but the cell derived from or developed from the nuclear cells only. One nuclear cell only developed to become the megaspore mother cell, and later this megaspore mother cell only go for meiotic division and finally produce four haploid cell at the end, and it is called as what the megaspore tetrad. Same thing we have seen here. This megaspore mother cell divide first meiotically and produce two cell mother cell dyad, then it go for the second division, that is equation division only, so it produce total four cell. All four cells are haploid in nature, right? This is what the things happening in the structure here. This is what, and one of out of these four haploid cell, three will degenerate and only one remains in the bud, and that only we call it as what functional megaspore mother cell. One of the nuclear cell only developed to become the megaspore mother cell, and this megaspore mother cell only go for meiotic division and produce total four cell at the end, and out of four, three cell get disintegrated, only one remain, and that cell is haploid in nature, and this haploid cell remains in this bud. At the end, we call it as what the functional megaspore mother cell, having only n number of chromosomes, right? Later, and this one undergo the division of its nucleus repeatedly, and division is mitotic division only, and the first division produce two cells, two nuclei, then second division produce what, four nuclei, and third division produce eight nuclei, right? And here you see structure. This is what functional megaspore mother cell, right? This is what the functional megaspore mother cell, and that only go for division of the nuclei. Only the nuclei will divide. First division of the nucleus produce two cell, two nuclei, and second division, and actually division is what the mitotic symbol, mitotic division only, and already the nucleus is haploid. Already its nucleus is haploid only. Here we have seen these are all what the haploid nuclei only, haploid nuclei. This is what the functional megaspore mother cell, and this functional megaspore mother cell is having what the haploid number of chromosome in the nucleus, and this cell only divide three times. That functional megaspore mother cell is having a nucleus having haploid number of chromosome. Remember, that will divide and produce two haploid nuclei. Only the nuclear division. Then after this first division, it go for the second division. So these two nuclei will divide into four nuclei. All the four nuclei are haploid nuclei only, and it go for the next division. So naturally, total 8 nuclei will be produced inside the functional megaspore mother cell. Functional megaspore mother cell, FMMC. Functional megaspore mother cell produce total 8 nuclei inside, and all these nuclei present inside the functional megaspores are having only n number of chromosomes. They are all haploid in nature, right? Then this is what the events happening here. This is what the first division of the nucleus of functional megaspore mother cell produce 2. Then go for the second mitotic division, produce 4. Then it go for the next mitotic division, third one, and produce total 8 nuclei inside the functional megaspore cell. And this 8 nuclei will arrange inside the cell in 3 places. And here it is what the micropyle N. In this micropyle N, 3 haploid nuclei will be placed, and central one is called as what the XL, and on either side what these 2 synergic cells. So total 3 cells are in the micropyle N. All cells are haploid only. And there are 3 nuclei arranged in the another end opposite to the micropyle. We call it as what the chalazal end. There we can find 3 nuclei, and we call it as what the antipodal cells only. And only 2 things remain. 3 are here in the micropyle, 3 are in the what you can say the chalazal end, and exactly the remaining 2 are placed at the center, and we call it as what the polar nuclei, right? So this is what the way through which what the ovule is getting developed, right? So now the functional megaspore mother cell, after the 3 successive mitotic division, and call it as what the embryo sac. And this is what the functional megaspore mother cell. It divides its nucleus 3 times. This nucleus is haploid, it divided 3 times and produce what the 8 cell, and all these 8 cells arranged in 3 different places. 3 are in the chalazal end, and 3 will be in micropyle end, and 2 will be exactly at the center, and later it is called as what the embryo sac. So embryo sac is actually developed from a functional megaspore mother cell only. And this functional megaspore mother cell developed from any one of the nuclear cell present in the buds only, right? So this is what the way through which this ovule is getting developed inside the ovary. And this topic is very important topic only. Let us see once again these various events happening during the process of what the megasporogenesis, right? And it is actually this megasporogenesis, actually this megaspore is formed just like what an outgrowth from the placenta of the ovary in the initial stage only. And later what happened, these buds undergo the repeated division and produce many cells inside, and we call it as what the nuclear cell only. And all the nuclear cells are diploid in nature. They are having the paired chromosomes in their nucleus. They are all diploid because it is an ordinary cell developed from the placenta only. Placenta is already diploid, and from the placenta only this bud has developed, right? So naturally it is also having what diploid in number of chromosomes, and later after a period of cell division getting a multicellular structure, it simply what you can say wake up from this placenta with the help of a stalk like a structure, that only we call it as what the funicule, later stage. And afterwards again this will divide repeatedly the cell present in this structure, that bud, then it produce what 2 integument for the protection by leaving a small aperture here, and we call it as what the micropyle only. And later what happened, it produce a megaspore mother cell, and one of the nuclear cell, it is all multicellular structure only. Among these, one nuclear cell, converted into megaspore mother cell, and that is also diploid in nature. And this megaspore mother cell divide meiotically and produce 4 cells. And all these 4 cells are haploid in nature. This megaspore cell will go for the first division, produce 2 mother cell dyad, and this mother cell dyad go for the next division of meiosis and produce what the mother cell tetrad. So there are 4 cells in the mother for tetrad, and this mother's tetrad, this is what the division here. This is what the 4 cell megaspore tetrad. It is what the megaspore mother cell developed from one nuclear cells only, will divide meiotically, and first division produce 2 cell, then go for the next division, produce what 4 cell, and we call it as what the megaspore tetrad, and it is called as what the megaspore dyad, right? And later, this one of these tetrad cell, megaspore tetrad, developed to become what the functional megaspore mother cell, and remaining three, disintegrated for produce, and three disintegrated, only one remains there, that only work as what the functional megaspore mother cell with a haploid number of chromosomes in their nucleus. And this cell, functional megaspore mother cell, go for its division of only the nucleus. Only nucleus divided three times, and division is what the mitotic one only. So every nucleus is having what haploid number of chromosomes. Total 8 nuclei will be produced after the 3 successive division. First division produced 2, second one produced 4, and third division produced total 8 nuclei, and all the 8 nuclei are haploid in nature. Same way, first division, second division, third division. First division produced 2 nuclei, second one produced 4 nuclei, and third one produced what? Total 8 nuclei, and all these nuclei arranged in the embryo sac in 3 places. One is micropylon, then central, and the chalazolan. 3 will be placed in the micropylon, and central located cell in the micropylon only we call it as what? The egg cell. On either side, synergid cells will be there, and 3 are there in the chalazolan. We call it as what? The antipodal cell, and the remaining 2 exactly at the center, we call it as what? The polar nuclei. This is what the embryo sac developed after the 3 successive division, right? This structure is called as embryo sac, and embryo sac only these 8 cells are arranged. See, 8 nuclei present in the embryo sac will be placed in 3 places. 3 places. 3 in what you can say chalazal end, and remaining 3 in the micropylon, and 2 exactly at the center, right? 3 nuclei in the chalazolan end called antipodal nuclei, then 2 nuclei at the center called as what? The polar nuclei, and 3, the remaining 3 at the micropylon only, and center nuclei in the micropylon is called as what? The egg, and 2 nuclei on either side of the egg is called as what? The synergids. This is what the structure we discussed while discussing about the structure of the embryo sac. See, arrangement of 8 nuclei in the embryo sac. This is what? The chalazolan. This one is what? The micropylon. In chalazolan, there are 3 nuclei placed, and we call it as what? The antipodal cells only. Later is getting the cellular structure, and these cells are having haploid number of chromosomes only, and the remaining 3 placed in the micropylon, and central one is larger called as what? The egg cell, and on either side we can find 2 more haploid cell, we call it as what? The synergids. And total 8, 3 are in the chalazolan, 3 are in the micropylon, and remaining 2 are exactly placed at the center, and we call it as what? The polar nuclei only, right? Right. This is what the come to the development of the embryo sac. This is what the detailed structure of embryo sac. The antipodal nuclei converted into antipodal cells. Antipodal nuclei, it is located in the chalazolan only, and nucleus of micropylon also converted into what you can say egg cell, and 2 synergic cells. On synergic cell on either side, and egg cell exactly at the center, right? And polar nuclei remains at the center as a nuclei, and we call it as what? The polar nuclei only. There are 2 nuclei. Sometimes it go for fusion to produce what? The central cells, and central cell is what? The diploid one. Otherwise, it is in the nuclear stage only at the center, and having what? Only n number of chromosomes. And see the structure of a mature ovule or egg. This is what the detailed structure we discuss, and this is what the funicule. I told you in the beginning, this funicule only attach the ovule to the placenta of an ovary. And by using the funicule only, it is absorbing all the nutrient from the placenta for the development as well as the maturation of ovule. Then here we can find there are two funicule for protection purpose. I mean, integument for the protection purpose. This is what one layer. It is what another layer. There are two integument by leaving a small gap here, and it is called as what the micropyle. Micropyle is a small aperture, a small opening where there is no integument cover, right? And inside so many cells are there. These are all diploid cells only, and this diploid cells present inside the integument is called as what the nucellus, and one of the nucellus only developed to become what the embryo sac. That development only we have discussed now. And this embryo sac, we can find there are 8 cell inside, and 3 are in the chalazalan. This end is called as what the chalazalan, and 3 are there in the chalazalan called as what the antipodal cells, and 3 are in the micropyle, and one is exactly at the center, larger in size, called as what the egg cell, and on either side we can see the synergids, and this polar nuclei located exactly at the center. Sometime here it is fused only. So total 8 nuclei or 8 cell present in the embryo sac, right? This is what this detailed structure of female reproductive gametes called as ovule, and we have discussed in detail about the process of what megasporogenesis. This is what the process of formation of female gamete from a bud-like structure developed from the placenta of an ovary. And this is very important. Even in the last pre-board examination also this question asked, right? And it is what very frequently asking question only. So whenever you explain the process of what the megasporogenesis or mycosporogenesis, you have to draw the diagram, and particularly the schematic diagram you have to draw, and you have to explain the detail of its development, so that it is very easy to explain by using such type of the schematic diagrams. Then come to the next event, pre-fertilization events. Now we are discussing about the event happening before fertilization only. First one is what? Gametogenesis, formation of microspore and formation of megaspore, male and female reproductive gamete production. Then second one is what? The pollination only. Already we discussed some of the details of pollination. Pollination is nothing but the transfer of pollen grain from the site of its origin to the stigma region only. And it required some kinds of agent for that, maybe biotic or it may be biotic, but some agents are very much required for this pollination. So transfer of pollen grain shed from the anther to the stigma region. After the maturation of what the pollen grain inside the anther, you know, this anther will go for breaking and taking all this matured pollen grain out, that will be attached on the surface of what this anther region. Whenever insect like what you can say that butterfly like insects sit on a flower for collecting the nectar, naturally what happens, this will stick on their body, and whenever they visit another flower of the same plant or another flower of the same species, naturally that pollen grain will stick on the stigma region of another flower, and this process is called as what? The pollination, right? So there are so many agents for this pollination in the nature. Some are insects, some are birds, some are bats. Then water and air can also act as what? The pollinating agents. And first thing, insect, birds and bats, these are all biotic agents, whereas water and air, these are what? Abiotic agents only. Biotic agent means what? The living agent, and abiotic means what? Non-living. For example, paddy, wheat, such type of plants normally use this abiotic agent like air only for the pollination purpose, whereas aquatic plants like Vallisneria normally prefer water as what? Agent for this pollination. But most of the plants, terrestrial plants particularly, preferring the living organism as a pollinating agent like insect, birds, or these bats. See, this is what the process of what? The pollination. Whenever these insects like this honeybee sit on a flower to collect the nectar, Naturally, from this male reproductive part, these pollen grains will attach to their body. You see the body structure of the insect having so many hairy appearance externally. So it can trap very easily these pollen grains on their body. Then come to this type of pollination. That self-pollination. It is a process of transfer of the pollen grain from anther to the stigma of the same flower or another flower produced by the same plant. These two are called as what? The self-pollination only. Self-pollination. Same plant or same flower, right? The pollen grain transfer to the stigma region of the same flower, or it is passed to the stigma region of another flower produced by the same plant. Both are considered as what? The self-pollination only. So self-pollination, just like what asexual mode of reproduction, never produce any variation in the coming generation, because both parents, I mean, these both are male and female reproductive gamete produced by the same parent only. That is why it's like exactly like what the asexual mode of reproduction. Then cross pollination is not like that. The pollen grains produced by a flower stick to the stigma region of another flower produced by another plant but the same species. So such type of what pollination is called as what? The cross pollination. Cross pollination definitely required an agent to transfer the pollen grains. That is called as what? The pollinating agents. Then come to type of self-pollination. Self-pollination, two types are there. Already we discussed this two type of pollination and self-pollination. One is autogamy. Autogamy means what? The pollen grains of the same flower stick to the stigma region of the same flower only. So both same flower only, male and female reproductive gamete produced in the same flower only. We call it as what? Autogamy. And diatogamy, here transfer of pollen grain produced by a flower to the stigma region of another flower but of the same plant. But effect will be same only. Autogamy and diatogamy, its the product will be showing the same kinds of character only, because only one parent is involved in this reproduction. Of course, it is sexual reproduction only, but only one parent is involved, because flowers are produced by same parent only, right? Then come to next one, that is autogamy, and some examples are mentioned here. And pollination is achieved in the same flower. Same flower. Pollen grains produced by one flower and that only stick on the stigma region of the same flower. And required synchrony in pollen release and stigma receptibility. The anther and stigma should lie close to each other so that self-pollination can occur. Required synchrony in pollen release. That means pollen grain and ovum should get matured at the same time. Then only it's possible for the autogamy. Normally, nature is getting developed these pollen grains and female reproductive gametes not at the same time. Right, difference is there. Male reproductive gamete getting matured little earlier than the female reproductive gametes to avoid this self-pollination or autogamy. The nature is having some defense mechanism here. Right. So pollination for autogamy, required a synchrony in pollen release and stigma receptiveness. Stigmas should be, what you can say, enough mature to receive that pollen grain at the time of its releasing. And anther and stigma should be lie very close to each other. That should be seen very close to each other, right? Then only go for autogamy. Then so that self-pollination can occur. It should mature and release in the same time. And these two things, male gamete and female reproductive part of the flower, should become very close to each other in a flower. Then example, that is what the clistogamous flowers, they do not open the flower. They do not open all the time. It is closed only. So here no chance of any kinds of cross pollination. Only what the autogamy is a method to pollinate a flower, because never it opened its flower and it produce assured seeds. 100% seed it will produce, because both male gamete and female gamete produced and get matured at the same time. So no way to, what you can say, waste their pollen grain. The entire pollen grain produced by the flower will stick on the stigma region 100%. So it is producing, I mean, it is offering what the assured seeds only, right? Then another one is what the cosmogamous flower. It is a bisexual flower, male and female gamete matured at the same time. It is what another flower, and both get matured at the same time. So that possibility is there for autogamy. Male reproductive part of the flower release the pollen grain. That time that female gamete is ready to matured and ready to go for fertilization. That receptibility of this stigma also there. So naturally, after releasing the pollen grain, in case of what the cosmogamous flower, it will attach to the stigma region of what the same flower, and it can go for what the autogamy, right? It is what the example for the clistogamous flowers only. And here you can see examples: oxalis, viola, then commelina. These are the three examples given in your textbook, right? And showing what the clistogamous flowers, and it is offering 100% seeds, assured seeds. And come to the next one, that is what the Giettonogamy, right? You know, Giettonogamy is different flowers of the same plant. It's exactly like what the self-pollination only. In autogamy, one flower, it is happening in same flower only, but here two different flowers but produced by the same plant only, right? Pollination achieved in different flowers of the same plant only. And functionally it is a cross pollination, exactly like cross pollination, because required an agent to transfer the pollen grain from one flower to another flower of the same plant. But it is exactly like what the autogamy only. It is similar to autogamy since pollen grains come from the same plant only. Male gamete and female gamete produced by the same plant only. So functionally it is like what autogamy only, because both male and female gamete produced by the same parent, right? Then it is another one. Here you can see the Giettonogamy and autogamy. And autogamy, same flower only involved. And here Giettonogamy, different flowers are involved, but is produced by the same plant only. So these two are self-pollination. One is autogamy and another one is Giettonogamy. You have to by heart the technical term used for these two type of self-pollination: autogamy and Giettonogamy. Autogamy involves only one flower, male and female reproductive gamete produced by the same flower only. But in case of Giettonogamy, two flowers are involved but produced by the same plant. Then here in case of inflorescence, you know that Giettonogamy is very, very common. Whenever this insect, suck the nectar from a flower, then it go to another flower, chances are there for Giettonogamy. Then come to the next one, that is cross-pollination. See, in cross-pollination, pollen grains are produced by one plant and ovule produced by another plant of the same species, right? So here some agents are very much required, and the agents only transfer the pollen grain to the stigma region of what flower produced by another plant of the same species. And it is scientifically termed as what the xenogamy. It is what the technical term for this cross-pollination, and it brings genetically different pollen grains to the stigma region. And it's what you can say, genetically different variations will be there in the next generation. Ultimately, the people are going for sexual reproduction, or individuals are going for sexual reproduction to produce variations in the coming generation. And it is possible only through what the cross-pollination, because here two parents are involved in the production of new generation. Two plants are involved in this what you can say process of reproduction. So naturally variations will be there in the coming generation, and it brings genetically different pollen grain to the stigma region and produce variation in the next generation. Here 100% agents are required. In case of what self-pollination, it may not required, and sometimes it's required, whereas in case of cross pollination it is very much required, an agent, biotic or abiotic agents. So such type of biotic agents are required, or sometimes abiotic agent also are required. So come to the agent for pollination, two types are there. One is what the abiotic agent, another one is what the biotic agent. Now we are discussing about the pre-fertilization events only. After discussing in detail about the events happening before fertilization, of course, we will go for a fertilization event, and also we'll discuss a post-fertilization event. These are the three things mentioned in your textbook. Pre-fertilization event, it includes what you can say, the gametogenesis for both micro gametogenesis and mega gametogenesis. And another one is what the pollination, and two agents are there for pollination, biotic or abiotic. Already we discussed. Abiotic agents are normally the wind and water only, whereas biotic agent includes what the insect, bats, then birds, etc. Right. Then abiotic, wind and water, these are non-living. And actually only a small portion of plant use abiotic agent. Contact of pollen grain to the stigma region is a chance factor in case of water and wind. That is why the plant, those who are going for using the abiotic agent as a pollinating agent, produce more number of pollen grains, because they will lose a lot of pollen grains during its transfer. Right, wind only carry, for example, paddy, it wind only carry the pollen grains. So naturally, chances are there to lose a lot of pollen grains while transferring. And these pollen grains are very light in weight also, so that it can be taken by the wind very easily. Right. And to compensate the pollen loss, flower produce large amount of pollen grains compared to the number of ovule available for pollination. The number of ovule available pollination will be very less, but it produces what more number of pollen grain. Why? Because during transfer, lot of chances are there to lose the pollen grain. Pollen grains are non-sticky and lightweight in case of wind pollination. Suppose in case of paddy, it is depending the wind only for the pollination purpose. And in case of such type of pollen grain, it is non-sticky and it is very lightweight. Then having exposed stamen, their stamen is very exposed, so that the wind can carry the pollen grains easily. And stigma is feathery, that receptibility is stigma only receiving the pollen grain, that will be like what you can say feathery type only to receive the pollen grain. And wind pollination is very common in grasses, and Vallisneria like aquatic plants are normally using water as a pollinating agents. And this question also asked once in the board examination, characteristic features of the plant use wind as a pollinating agent, and always it produces number of pollen grains, and these pollen grains are normally non-sticky and it is lightweight. And these plants are also having what and well exposed stamen, and to receive that pollen grain from the wing, it's having what you can say feathery type of a stigma. Right, you have to by heart 2-3 example for such a plant use wind as a pollinating agent. And this is what see that stigma region, it is just like what feather, right? It's so that it can receive the pollen grain from the atmospheric air. And see, this is what the male reproductive part. So it is well exposed, so that wind can carry the pollen grain easily to the stigma region of another plants. Then water as a pollinating agent, a common in very algae, bryophyte, pteridophytes, Vallisneria, hydrilla type of plants, hydrilla or Vallisneria. It is commonly using water as a pollinating agent. In Vallisneria like plants, pollen grains are released to the water surface, and which are carried out by the water current only to the female flower for fertilization. Vallisneria, here you can see the Vallisneria. Here, this is what the Vallisneria. See, Vallisneria, it is what the staminate flower, and it is producing the pistillate flower. Staminate flower means what the flower consists of only male reproductive part, and pistillate means what only the female reproductive part. And these pistillate flowers, usually float on the surface of the water only, and staminate flower is getting developed below the water surface. And after releasing this, what the uh, male reproductive gamete from the staminate flowers, it will go to the surface, and according to the water current, it will be taken, and it will find what the female flower normally float on the surface of this water only and attach to the stigma region of the pistillate flower. This is what the way through which the Vallisneria is pollinating its pollen grains, and it produces two type of flowers here. One is what the staminate flower. Staminate flower means what the flower produce only the male reproductive gamete, and pistillate means only the female reproductive part present there. And normally it is keeping the pistillate flower on the surface, whereas the staminate flower below that. After producing male gamete, pollen grain by the staminate flower under the water, this will move up because it is very lightweight, and it will be taken to other part through the water current only, and it find the pistillate flower and stick the stigma region of this flower and go for pollination, right? Then seagrass is also pollinating by using this water only, right? Then come to the biotic agents. Biotic agent means here insect like, bat like, or bird like individually used as what agent for pollination, biotic agent for pollination. See, majority of flowering plants are using these animals only as an agent for pollination. Flowers of animal pollinated plants are specifically adapted for a particular species of animal, and it is species specific. Most of the plants are very specific to a particular species, so that it never visit what the plants of another species. So pollination is very easy in case of such things, because these are all species specific. A particular species of butterfly visit only a particular species of plant for collecting the nectar. Whenever it visits but the same species for collecting nectar, naturally and accidentally the process of what pollination will happen. So insect, birds, bat, these are a common example for this biotic agent. You see, while collecting the nectar from a flower, you see that the body face, actually the face of what? An insects only, butterfly only. How much pollen grain stick on the facial region of what you can say, this insect. And whenever this insect visit another individual, another flower for the same purpose, this pollen grain attached to their body will stick on its stigma region. So accidentally the process of pollination will take place. Then come to the characteristics of insect pollinating plants. We have discussed in detail about the characteristic features of the plant, those who prefer wind as a pollinating agent. Now we are discussing about the characteristic features of what the plant use insect as a pollinating agents. So here flowers are very large in size. Normally plant produce a large, colorful, and what you can say, produce fragrance, good smell, and rich in nectar. Actually, plants are giving nectar as a reward to the pollinating agents, because actually the agents are visiting a flower for collecting the nectar. The purpose is to collect the nectar only. So actually it is a reward given by the plant for this pollination purpose. So normally the flower produced by the plant for insect pollination are larger in size, and it is very colorful and attractive. Normally day blooming flowers, it's very colorful. Maybe it's what, red, or what you can say, green, yellow, I mean blue, yellow, like that only, and also produce very good fragrance and rich in nectar. The night blooming flowers, normally it's white in color. Why? Because it need a contrasting color to attract these nocturnal insects. Then when flowers are small, number of flowers clustered to form an inflorescence. You maybe notice many inflorescence, right? Here, this is what the inflorescence. I've seen that the inflorescence here. See here, it is what the inflorescence. It is colorful, but the flowers are very small. Whenever it is small, normally it is in form of clusters only, and that only we call it as what the inflorescence, right? Right. Okay. So we will discuss that. I think is your time is over now. Now you have to go for a breakfast. So we will have a discussion on the characteristics of insect use the pollination agent, acting as a pollination agent in your next section, and next section start from 2:30 to 4 o'clock, only 1.5 hours. And you have 3 section in day, in morning from 6:30 to 8 o'clock, and another one for half an hour only, 2 section, 2:30 to 4, and another one is from 6:30 to 8 o'clock in the night. Okay, we will see you later after in the second section and discuss the characteristics of the insect pollinating agents helping for pollination plants. Okay, thank you so much.